Okay, so here's what's going to happen this week for the bio lessons. I'm going to break down each organelle into their own video so that you can study each organelle individually. And then at the end of all of those, then I will upload an assignment that covers all of the organelles for the eukaryotic cell. But I don't want to do them all in one video. I want to do each one individually. So I'm going to start with the plasma membrane or the cell membrane. Now, there's two main functions for the plasma membrane. The first one is to act as a barrier between the inside and the outside of the cell. And the second one, which is probably, well, not just as important, is that it controls what substances can pass into and out of the cell. So we describe the cell membrane as being what we call partially permeable. So let's break down what that means. Permeable means to allow substances to pass through, and hopefully we know what partially means. It's like some of or a part of. So being partially permeable, or sometimes is called semi-permeable. But the word semi means half, and it doesn't necessarily allow half of the things to come in. So this is the preferred terminology, partially permeable, which basically just means that it allows some of substances to pass through the cell membrane or the plasma membrane, but it does not allow everything to go through. Oops, let me get that away. It is made mostly of proteins and lipids. By far, lipids are the #1 component of the plasma membrane, followed by proteins. And then, as I said in a previous video, there's some cholesterol that's in the lipid bilayer, and there's also some carbohydrates that are attached that serve either as a binding site for things to attach to the cell, to maybe like a hormone or another cell attaches, because cells of course have to be attached to each other, or as just an identifier as this cell belongs to this body. Okay.
This is a cell membrane. The cell membrane separates what is inside the cell from what is outside the cell.
Okay, great. Think I'll be deleting that because that was pointless. All right, so here is where I want to focus on the structure of the cell membrane and focusing on the phospholipids. So as a review, the phospholipid is the main type of lipid or fat that makes up the cell membrane. The phospholipid has a polar head, which is hydrophilic, which means it loves water. And we know that water is also polar. We have to remember that water is most of the outside environment and water is most of the inside environment. So you can see from the diagram here, you need the heads to be facing both outside and inside. Now, the other half of the phospholipid are these nonpolar tails. Because they're nonpolar, they're hydrophobic, which means water fearing. Hydro means water, phobic means fearing. If you have a phobia, that means you have an intense fear of something, and then the phobias are defined by the prefix or whatever it is. So for example, for a long time, I suffered from something called aerophobia or aviophobia, which is a fear of flying. I'm fine now, but so because this end of the phospholipid does not like water, and it will automatically orient itself away from water, just like oil and water, because oil is nonpolar. So nonpolar and polar do not mix, like oil and water. So imagine if you didn't have a 2-layer system where the nonpolar parts were protected in the middle away from the water, what would happen to the cell membrane? It would be just like oil and water. So all of the contents of the cell would be separated from this lipid bilayer, and like the cell wouldn't be able to take shape at all. So the phospholipids are a huge part of the structure of the lipid bilayer. So there's 2 layers of phospholipids because the tails of the phospholipids are hydrophobic. They're nonpolar. Okay, so here is another, a different diagram of the membrane lipids. You can see that there's the green and the blue, but their tails are all protected on the inside. Again, the specific lipid here is called a phospholipid. And again, the polar head, which you see here, is hydrophilic, meaning water loving, and the tails are hydrophobic. So here is a really good example of what happens if you have a non-polar substance that's in water. It doesn't mix at all. It completely doesn't like water at all. When you take it out of the water, okay, it's all fine. But imagine if you had only one layer of lipids, like it would be like that, because again, most of the inside and outside environment of the cell is aqueous, it's water. So you need to have the outside and the inside parts of the membrane that are okay with being around water. Okay, so then the next part of the cell membrane are the proteins. So there's 2 types of proteins that I've mentioned before. There's some that go all the way through from the inside all the way to the outside. These are called integral proteins. Integral means, just as a regular English vocabulary word, integral means to be a really big, like, trying to think of the right word. It's like when you say something's an integral part, it means it's a necessary, like it's built in. That's a really good way to describe it. It's built in, a built-in part of a whole. So the integral protein is a built-in part. It literally extends from the outside to the inside of the cell membrane. So we call it embedded in the lipid bilayer. It doesn't mean it's attached to one side or the other side. It's embedded. It's like right in the middle of the bilayer. So these are the proteins that often have a carbohydrate attached, which you can see here, as I've explained before, what the function of the carbohydrate can be. It can join cells together. It can also be a receptor site. An interesting fact about how carbohydrates function as receptor sites, they're also receptor sites for viruses like coronavirus. Coronavirus attacks mostly the respiratory system. There are receptor sites on I think they're the cells in the lungs. And there's just certain carbohydrates. You remember from a previous video, I talked about how proteins are completely based on their shape. Their function is completely based on their shape, like a lock and a key. So the protein has to match a particular shape. So it turns out that a lot of our carbohydrate markers on the outside of our cells are a perfect match for a lot of viruses, including like HIV attaches to a particular type of white blood cell, coronavirus, a cold virus, a flu virus, all of viruses, they have proteins that can match the shape of our carbohydrates or other proteins on the surface of our cells, which is how they're able to attach and then inject their DNA, and then they copy themselves, and that's how we get sick from it. The biggest function of the integral proteins is that they act as a channel for substances to go in and out. So really quickly, talking about the lipid bilayer and why the cell membrane is called selectively permeable, you have some substances that can go straight through the lipid bilayer. Now, because the tails are nonpolar, only nonpolar substances can go straight through because they have to be able to go through those tails. If you have a polar substance, it's not going to get past those protected tails on the inside. But things that are non-polar, like oxygen gas can go straight through the lipid bilayer. But if you have something like carbon dioxide or glucose, other things, ammonia is a big one. Ammonia is a compound that's formed when your cells break down proteins during metabolism, and ammonia needs to be able to get out because it's very toxic to the cells. So it has to be converted in the liver. So all the ammonia, when your cells break down proteins, it produces ammonia. The ammonia goes to the liver and gets converted to a less toxic compound. But ammonia is a very polar molecule, so it cannot go through the lipid bilayer. It has to go through an integral protein. So the other important thing to understand about channels as far as the cell membrane goes is that channels are, because they're proteins and because proteins have a specific shape, which is dependent on their function, or their function is dependent on their shape, every substance that gets transported through a protein has its own unique protein in the cell membrane that it can go through. So for example, let's say this protein here is for ammonia. Only ammonia can go through that protein. If you have carbon dioxide cannot go through that protein because the shape of that protein is for ammonia and ammonia only. Okay, the other kind of membrane protein is called a peripheral protein. Now, peripheral means on the side. So if you want to test your peripheral vision, you stare straight ahead and you can like take your fingers and you move them to the sides like that. And the point at which you can no longer see your fingers, even when you're looking straight ahead, that's the limit of your peripheral vision. One of the tests that you do at the to get your driver's license is a peripheral vision test where they blink lights and they ask you to say what color it is to test to see if you can, if you're driving straight, you can see that there's cars coming from either way. That's one of the tests. Okay, now the model that we use to describe the behavior of the cell membrane is called the fluid mosaic model. Now in biology, we have, or like in just in science in general, you start with a hypothesis, right? And you test the hypothesis and you get evidence to back up your hypothesis, whether it's correct or not. If your hypothesis is correct, you can then form a conclusion. And based on other evidence, you can form what's called a model. And a model is something that you can use to try to predict behaviors or outcomes in the natural world. And this is the model that we use to describe the behavior of the cell membrane. You can see from the graphic that I have there, this is what I mean by fluid mosaic. There's two parts to this, the fluid part, the mosaic part. Now, the mosaic part, if you know what a mosaic is a large picture that's been made out of smaller pieces. Oops. Let me just show like an example of a picture. Okay, so that's an example of a mosaic. So it's a picture that's been made out of smaller pieces. You can see the smaller pieces there, if you didn't know what a mosaic was already. So your lipids, your two types of proteins, your carbohydrate chains on the top, your cholesterol in the middle, all of these are the pieces that make up the larger picture of the cell membrane. The fluid part comes from this animation that you're seeing here. You're looking at integral proteins, which are able to move laterally, meaning side to side. They can move within the lipid bilayer. They're not stuck, they're not stationary or static inside the membrane. They can actually shift around. So that's the fluid part. The lipid bilayer acts like a fluid, so lipids and proteins can move laterally within the bilayer, and the pattern or mosaic of lipids and proteins is constantly changing. That's the fluid mosaic model.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.